In this video, we're going to go over some steps for fatty acid synthesis and just some enzymes and some short steps just to connect things and make things make a little bit more sense. This pathway is not as straight and linear as the other ones have been. There's a lot of jumping around, a lot of intermediates that can be used in various places of the cycle. So this is the first time we're kind of seeing things fold together as one, not separate things going down to one another, but we're going to start feeding back on each other. So I'm going to start off first with citrate. Citrate, I'm going to make an arrow all the way out here. And this is going to represent it transporting to the cytosol. So the Krebs cycle takes place in the mitochondrial matrix. So citrate is in the mitochondria right now, and we're going to transport it to the cytosol to continue this pathway. So with this, we are going to turn it into oxaloacetate, which oxaloacetate is used in so many things. We used it at the beginning of the citric acid cycle. We ended with it in the citric acid cycle, and remember, it is also that intermediate for gluconeogenesis. So from here, also make acetyl-CoA as well as NADPH. And this is our fatty acid synthesis. We'll talk about more how acetyl-CoA works into fatty acid synthesis later, but for now, just remember that acetyl-CoA goes with fatty acid synthesis. And so acetyl-CoA is also up here. So I'm going to make a note that this also has to do with fatty acid synthesis. So this isn't as specific as some of the other pathways that we have done. This is just kind of trying to get a bigger picture of where these intermediates fit in with one another, where the synthesis steps fit in, and just some of the overall big picture things. It's also important to note that oxaloacetate can be turned into malate, and we oxidize NADH in the process, forming NAD+. Now, malate, if you remember, is a part of the Krebs cycle. It is an intermediate here, right before oxaloacetate. So this is that same reaction, malate to oxaloacetate. And we use malate dehydrogenase, so I'm going to mark that up here. And now it is also helpful to note that this oxaloacetate can also go through that exact same reaction, form malate with malate dehydrogenase. Now, if we zoom out, the last kind of overarching step that we can draw here is that malate has the ability to go all the way back to pyruvate. And in this reaction, we are reducing an NADP to an NADPH. So this overall might seem a little discombobulated, but it's just important to know some of these key facts in how they all interact together with fatty acid synthesis. So acetyl-CoA plays a big role in fatty acid synthesis, but we need to remember that the citrate that starts this all needs to be transported to the cytosol for all of this to happen. And we can also see here that oxaloacetate and malate readily get converted to one another with malate dehydrogenase, and that can be seen all throughout the biochemical pathways. It can be seen in gluconeogenesis, it can be seen down in the Krebs cycle, all the way down here. So these are just important key ideas to remember. The notes for this will be in the link tree. It will be biochemistry and then fatty acid synthesis. But that is it for this video.